This is a lecture on regulation of gene expression in bacteria from April 4th, 2020. And the main goals of this lecture are to define some different terms for genes, based on how they're expressed, as well as talk about positive and negative control of transcription and the roles of several different proteins in this process, including activator and repressor proteins. And so genes can be expressed at different levels, and this control of gene expression can happen at many different levels as well. And so the part that we're going to be focusing on here is actually the transcriptional level. And so we're really just going to talk about how genes can be regulated at the level of making mRNA from DNA, or at the transcriptional level. But it is important to note that they can be regulated both at the level of translation, or the level of making a gene into a protein, as well as after the protein is already synthesized in a post-translational way. And so coming back to transcription, which is the part that we're going to focus on, there are some genes that need to be transcribed or that are needed all the time. And these genes that are continuously expressed because they're necessary all the time are known as constitutive genes. And sometimes they're also referred to as housekeeping genes because they tend to maintain all the processes necessary for a cell or bacterium to survive. And so they sort of keep the house, or they keep the cell running, and therefore they need to be constantly on or constantly expressed. But there are some genes that are only needed at certain times or under certain specific circumstances. We all know that making a protein requires a lot of energy and input, and so if you don't need a protein at this particular moment or under these particular circumstances, it makes sense to not make that protein. And so one of the types of genes that's only made at a certain time is known as an inducible gene. And an inducible gene encodes a protein whose level increases in response to a small molecule, or that's made in response to a molecule, which is known as an inducer. And there are some genes that encode proteins whose levels decrease in response to molecules, and these are known as repressible genes, or genes that can be turned off, whose protein levels will go down in response to a small molecule called a repressor. So there are a lot of the proteins that are required to regulate the initiation or the start of transcription. And these regulatory proteins are relatively different from each other. They're pretty diverse in their structure and how they look. But most of them contain DNA binding domains. And the reason for that is because most of them bind to DNA and then recruit other proteins to start transcription, right? And so transcription is a process of taking DNA and converting it into RNA, and in order for that to happen, certain proteins need to recognize that promoter region before a gene, and that's generally what regulatory proteins in this context do. And so in order to bind the promoter, they need to contain a DNA binding domain. And one of these proteins that regulates sort of the start of transcription are known as activator proteins. And these are proteins that can bind the DNA at specific sites and promote initiation or the start of transcription. They bind to a specific region, and then the promoter, which you can see in light blue, that's known as the activator binding site. And activator proteins are associated with positive control of genes or turning transcription on. So as you can see here, you've got this activator protein shaped like New Jersey, which can bind to this activator binding site and cause the recruitment of the transcription machinery, the RNA polymerase, and transcription can be positively controlled or turned on. And alternatively, we have a protein who, when binds to DNA, actually inhibits the start of transcription. And these proteins are known as repressors. And repressors bind to a specific area of the DNA, which can be seen in yellow. It's called the operator. And they're associated with negative control mechanisms, or turning off of transcription. And so when a repressor protein is working, it's turning transcription off. You can see this repressor here binding to the operator in yellow, and transcription was going along just fine, but when that repressor is there, transcription gets turned off. Now there's also an additional level of regulation of the regulatory proteins themselves. So in addition to activators and repressors being able to turn on and turn off transcription, they can in turn be turned on and off themselves, right? So an activator protein and a repressor protein exist in both an active or off state and an active state or on state. And the way that they are regulated is in a way known as allosteric regulation by different effector molecules. And what allosteric effectors do is they bind somewhere on a particular protein and they force it to change shape. And usually they can change it from an active conformation into an inactive one by changing its shape, or they can change it from inactive to active. And that's because a protein's function is dictated by its shape or structure, right? And so allosteric molecules can change the shape and therefore change the functionality of these different activator and repressor proteins. I'm going to start by talking about some of the allosteric effectors of activators. And so normally you have an activator that is in its off state, not bound to the DNA. And there is a molecule or an effector known as an inducer that when it binds to this activator, turns it on so that it can bind to the activator binding site and turn transcription on. And alternatively, if we have an active activator protein already bound to the DNA, already doing its thing, there's an allosteric effector known as an inhibitor, which can bind to the active activator, cause it to change shape, and now it's unable to bind to the DNA, and transcription turns off. Repressors also have allosteric effectors, and so in a normal state, a repressor is off, and then an allosteric effector called a co-repressor can bind to it, change its shape, and allow it to now bind the DNA and do its job of repressing or turning off transcription. And alternatively, a repressor that's already active and bound to the DNA can be inactivated by the binding of another allosteric molecule called an inducer. And that inducer binds to this active repressor, changes its shape, and turns it off. And if a repressor is not doing its job, transcription occurs. And so what I would like you to spend some time doing is thinking about these different proteins, both activators and repressors, as well as all of the allosteric effector molecules. So inducers, inhibitors, and co-repressors, and their function in the and how they end up turning on or turning off gene expression. And for the purposes of your exam, I would have suggested, if it was closed book, to spend a good deal of time on this, because the concept can be kind of difficult. There's a lot of double negatives, and there's a lot of different outcomes. But one place that you can kind of use for your open book exam is this figure here, which is figure 14.3 in Prescott.